Hello everyone, my name is Mohd Arish Usman and I'm in my third year of PhD. My PhD project is related to engineering biochar-based fertilizers to address phosphorus stratification issue in Australia. So what is stratification? So in the morning talk, Professor Richard Bell has told around 90% of the Australian farmers nowadays are growing crops in no-till lands. In no-till lands means whatever the fertilizer that we are putting in, it is not going to the subsoil zones where the plant roots are. So if the crops are not receiving these it's not absorbing these nutrients, it reduces the fertilizer efficiency also, and also stunted growth is there. It means we are providing more fertilizer as compared to the growth is less. So how can we reduce the stratification in soil? A lot of fertilizer management strategies available in Australia where people are doing the crop rotation and also they are bending fertilizer deep into the soils. But is there any strategy where we can just put the any fertilizer into the topsoil into the 1 to 2 centimeter zone, and it can also absorb, it can also show the controlled release of these nutrients, and also the nutrients will go into the subsoil zones. So for this project, what I'm using is I'm using coffee ground waste as my biochar material. As you know, around 83,000 tons of coffee waste is generated per year in Australia only. So this is directly going to the landfills, and also, so if we utilize this coffee waste grounds to make some biochar-based fertilizers, we are also addressing the waste of agricultural waste which is generated in Australia. Because we have done a lot of characterization, also see the release profiles, and is showing superb results. So we are in future we're planning to apply for an IP. So that's why I can't show you the whole process how I'm synthesizing the material. But this is the fertilizer, the final fertilizer. This is just one of my sample, where you can see the phosphorus is uniformly distributed to the biochar matrix, and also we are loading zinc into it. So what zinc is helping? It is a micronutrient. It helps in the early root proliferation of the crops such as wheat, barley, and also, and if any grower or farmer wants that we don't need zinc, we want manganese, or how much manganese, we need 3% manganese, can we load it? So the answer is yes. Through this strategy, we can load any nutrient into the biochar matrix. Lastly, because seeing is believing, until we do the experiments and the farmers are more interested in seeing the crop growth. So this is the growth in just the soil only. This is negative control. Okay, this is the growth with just soil. This is the wheat growth in 28 days. Then we applied then we applied the generic fertilizer MAP, which is usually used by many Australian farmers. When we go to the field trips, they will use MAP or DAP to produce canola, wheat, or different crops. So when we put the MAP into the topsoil, this is the growth that is coming in just 28 days for wheat. But then we also apply that MAP in both the topsoil and the subsoil. As you can see, the growth is enhanced when MAP is also put into the subsoil zones. Then this is the growth of the wheat that is coming with my fertilizer that we put it into the. This is just one of my sample. As you can see, there's a enhancement in plant growth and height you can see, and also the leaf height. So this is the conclusion of my project. Until 28 days, we can see that the average plant height increases from, with the commercial fertilizer, the plant height is 20.25 centimeters. And with our material, the plant height increases from 20 to 31.5 centimeters, which is quite exceptional within 28 days of the study. And also the leaf side, now we are also seeing the stratification. We are also collecting the leachate from different soil layers. Until now, we have done it from the 10 centimeter layers. So we can't just conclude that it is also helping the stratification also. Lastly, I would like to acknowledge my Professor Ajay Amino, for his constant support and guidance throughout my PhD project, to the whole soil CRC team, and also our co-supervisor, Dr. Lucas, and Professor Richard Bell, who is helping us with the plant pot experiments, and Maren, who is doing all the plant pot experiments for us. And also we are looking for future collaborations where people can help us with the extensive characterization such as the 33P isotope, or anybody can help us with the phosphorus diffusion with the synchrotron and everything. We are happy to collaborate with them. Thank you.